Hello student, today in this video I'm going to discuss about the Mendel laws of inheritance. In last class we have discussed about the contribution of the Mendel, the father of genetics, Mendel law of inheritance. After performing experiment on pea plant for 7 years, he had given 3 important laws. The first law is law of dominance. Let's try to understand, okay, the law of dominance. Now, if I say monohybrid cross, monohybrid cross. It means the cross between two different plants, okay, having one contrasting character. For example, tall, short plant, okay. So that is said to be monohybrid cross. For example, the law of dominance or the Mendel laws of inheritance, it always starts from the pure line, okay. So this is a pure line, a tall plant having allele as a tall plant, capital T, and a dwarf plant, a short plant, that is small t, small t. Law of dominance always starts from the pure line. It means that you can see their allele, capital T, small t, small t. Just say that this is tall plant and this is dwarf or short. Now, during gamete formations, this is capital T, this will be capital T, this will be small t, and this will be small t. Let's make Punnett square. Punnett square. This is a method of probability to find out the character of an individual. So, this is capital T, small t, small t. So if you cross it, it will be capital T, small t, capital T, small t, capital T, small t, capital T, small t. Okay? So you will find that the phenotypic ratio means if there is a capital T, small t, the character of that plant will be tall. So this one also, capital T, small t, will be tall. This one also same, and even for this. You can see that the phenotypic ratio is completely tall plant. It means that if there is a dominant allele, okay, the character will be dominant. Here the dominant character is tall, right? So the plant will be tall. So its phenotypic ratio will be 4:4. In the first generation, we are talking about the first generation, okay? Now if you see the genotypic ratio, even the genotypic ratio is 4:0. Why? Because you can see that capital T, you can find 4 same allele, right? Capital T, small t, 1, capital T, small t, 2, third and 4. That's why I'm writing as 4:0. So this is said to be the law of dominance. The next is about the law of segregation. When we say segregation, it means separate. Segregation means separation. Okay? Just say that this is a tall plant, capital T, a homozygous dominant plant, okay? And if I take as capital T, small t, heterozygous dominant plant, okay? Now, during gamete formation, capital T, small t, right? Capital T, small t. Now, this plant is a tall plant, homozygous dominant plant. We can see their allele is also having capital T, a dominant allele, right? If you see this as a tall plant, dominant, the character is, it's a heterozygous dominant plant because T is dominant. You can see that during gamete formation, I'll write here, gamete formation. During gamete formation, this capital T is a dominant. So for that plant, it will be pure tall plant. And for this also, it will be a, for that particular plant, this, even though there is a small t, it will be a pure short plant. Okay, so this is said to be the law of separation or law of segregation. Okay, and the law of segregation is also said to be the law of purity of gametes. Purity of gametes. Okay, the next third law, this is the second law. The third law is law of independent assortment. Law of independent assortment, and to understand that we have to understand dihybrid cross. Law of independent assortment. And to understand this law, we have to take an example of dihybrid cross. Now, what is dihybrid cross? The crossing between the two character. Okay, just say capital T, capital T. It's one character. Its contrasting character will be small t, because it will be a dwarf, right? Just say that. I'll take another character. For example, purple. Capital P, purple. Small p, small p. So it can be white color. So a tall plant with purple flower, okay? Just say a short plant with white flower. So this is an example of dihybrid cross. Now let's take an example of, from your textbook, another example was given, that is capital R, okay? And capital Y, means round yellow. Round yellow. Okay, so small r, okay, wrinkle, small y, green. So always know that the law of the Mendel laws, it always starts from a pure plant, pure line. Okay, so this plant having completely homozygous allele, capital R, that represent round, and this capital Y represent yellow, and these small r's represent wrinkle. See, this is pure line because it is wrinkle, because it is showing homozygous allele. Okay, and this is small y. That's why I will consider this as a pure line of dihybrid crosses. Okay. Now during gamete formations, this R will come, and this Y will come, and this small r will come, and this small y will come. So such type of gametes, okay, we will produce four same of this type of gametes will be produced. Okay, now it will go for, this is during the F1 generations. Just say this is F1 generation. Okay, during F1 generation, let's see, with the help of Punnett square, what will be the character of that plant during F1 generation? Capital R, capital Y, small r, small y. So capital R, Y, R, Y. So if they crossed with capital R, Y, right? Capital R, small r, so capital R, small y. Same as this, R, Y. So just say that this is capital Y, small y. So this is capital Y, Y, R, y, small y. Okay, so this is capital R, small r, capital Y, small r, R, Y, R. Okay, so this is capital R, small y, capital Y, small y, capital R, small y, capital Y, small y, small y. Okay, so with the help of Punnett square, we can make like this type of contribution. Okay, now if I make it self-crossing, okay, we got as capital R. Just say that capital R, capital R, capital Y, capital Y. So small r, small r, small y, small y. So this is R, this is Y. So this is R, this is small y. So by fusion, capital R, small r, capital Y, small y. Okay. Now, if I go with crossing during the gamete formations, then this one and this one will combine. So this is capital R, capital Y. So capital R and small y again, capital R, small y. Okay. Then small r, capital Y, small r, small y. So this will be the gamete formation, the F1 gamete formations. Okay, it means that you're going to get round yellow, round wrinkle. So during gamete formations, we will get this type of products. Let's go for selfing now. Capital R, small y. I want all the students to look properly. Capital R, small y, to understand this concept, to understand Mendel's laws of independent assortment. So this is capital R, capital Y, capital R, small y, small r, capital Y, small r, capital Y. So same as like that, you have to make like this: capital R, capital Y, capital R, small y, capital R, small r, y, then R, Y. This is capital. Okay, we make the same here, and for that also the left hand side we make the same letters. Okay, now let's cross it. Capital R, capital R. So capital R, capital R, capital Y, capital Y. I'll show you one. Okay, capital R, capital Y, small y. Capital R, small r, capital Y, capital Y. Capital R, okay, capital Y, small y. Same as like that, you have to make the Punnett square. Now, let's discuss this. Capital R means what? Round. Capital Y is yellow. It means that this plant have a character of yellow and round. Same as like that here. Capital R, it will be round. Capital Y, small y. So dominant character, the law of dominance. So the plant will show, even though there is a Y, heterozygous, this is a heterozygous dominant allele. So capital Y will always show yellow color. So capital R, small r, same thing. Even it's a dominant, heterozygous dominant allele, so it will be round. Capital Y, it is yellow. So capital R, small r, even though it is a heterozygous dominant, so it will be round. So this is a heterozygous dominant allele. So capital Y is there, so it will be yellow. So the law of independent assortment, for dihybrid process, after crossing, independent assortment, after crossing, after self-crossing, okay? The F2 generation, the genotypic ratio will be, the phenotypic ratio will be 9:3:3:1. So this is the phenotypic ratio. Phenotypic ratio. So these three laws was given by Mendel, and till today in the molecular biology we are using this basic law. In the next class we'll discuss about the sex determination in human being. Thank you.